because I said what was on my mind. I made a lot of enemies. I made a lot
Welcome, everyone, to Science from the Fringe. My name is Bryce Nickels. I'm a professor of genetics at Rutgers University, and once again, I'm joined by distinguished virologist Simon Wain-Hobson for the third installment of In Defense of Virology. Simon, how are you doing today?
Fine, thank you so much. We got a bit of a storm outside, but apart from that, inside with you, great.
One of the issues that I think we've been discussing in terms of how to structure these episodes is explaining what In Defense of Virology is about. So I did take the liberty of asking ChatGPT to come up with some taglines for this show, if you don't mind. First, do you have any suggestions for a tagline that would quickly communicate what In Defense of Virology is all about? Not to put you on the spot.
I'm trying to find something succinct. You can go a long phrase. You want a quick tagline. Getting it right.
Getting it right, which was the title of one of your essays. So yes, right. Yeah. See, wow. I follow this stuff. Now, I had put something. I had defending virology from itself, one episode at a time. That's what I had put before. But then here's a couple more taglines: exploring how modern virology has lost its way and how to bring it back. Another is where science meets conscience, or when virology forgets its purpose, truth becomes a casualty. And finally, maybe this is the one: look, it might not be great, but at least it's better than This Week in Virology. I don't know. What do you think of that?
No.
Okay. We could come back to this, Simon, if you want.
Yes. When there's too much spin, truth becomes a casualty. Yes, it's true. I mean, everyone knows the term spin doctoring, and in politics it's normal. But if this happens in science, then truth is a casualty. Clear, no question.
One of the topics that we've covered in the first two episodes of In Defense of Virology is the concept of informational hazard. And you wanted to go over two specific examples of these types of informational hazards today.
Yes. So you have the virus. I know as a virologist, we'd be working in the lab in biosafety hoods, and we'll be working physically with the virus. And that's what we do much of the time. But you can take the genetic information of a virus, and you can synthesize the gene. You can actually, these days now, synthesize the whole virus. And so with just the information, as mentioned last week, I could send you the sequence of a new virus that we had discovered and not yet published. And because we were working together, I would send you that sequence, and you could make the virus in your own lab in probably 3 weeks, 4 weeks at most. So I wanted to take two examples to push this to help listeners understand. So we're going to talk about two viruses for which we don't have virus, but we pull it back through genetic information. So everyone has heard about Spanish flu. This is the virus that broke out in 1918. And in fact, because it's called Spanish flu, people might think it was in Spain. The first documented case was in March of 1918 in Haskell County, Kansas. And that from there, we was turned up in France, Germany, the UK, and spread around the world. And of course, it was the end of the war, for World War I, and no one wanted to sort of say, well, this came from the US, and because this was not very good. They wanted to find a neutral term. And of course, Spain was not part of the First World War. And the King of Spain got flu. And this was talked up because the King of Spain has flu. And it was because of that it ended up being referred to as Spanish flu. But as mentioned, the first case was in Kansas. And you wouldn't call it USA flu, it's called Spanish flu. Now, this virus killed with all sorts of bacterial complication, and there was the end of the war. Lots of people, the troops caught it. Lots of people in Europe caught it because after the war there was even more poverty than before. And it's estimated something like 50 million people died in a population of 1.8 billion, something like that. It's estimated the third of the world's population was infected, but 50 million died, which is phenomenal. Just to normalize it, COVID is estimated to have killed maybe 20 some million people, and our population is much bigger than it was then. So Spanish flu was something really nasty. And it moved on, it became seasonal flu. Then it was replaced in 1957 by what was called Asian flu, which was replaced in 68 by what was called Hong Kong flu. And Spanish flu came out at a time when we didn't have modern molecular genetics. So from a virological point of view, it was curiosity. Well, what was it? And it would be nice to have the information. Nice to have in a scientific sense, in a virological sense, because it was the biggest pandemic of the last 100 years. So researchers tried to get hold of the material, and of course the idea was, well, let's try and find cadavers that were kept cold in the permafrost. And a number of attempts were made, and finally a group that was supported by the NIH, a CDC and NIH team, found cadavers, I think, in Alaska, and they were able to, using this ultra-sensitive method called PCR, and we've mentioned this before because this is that rapid test that was done on street corners during COVID. Very, very sensitive. You can pick up tiny amounts of material and amplify it a million, a billion fold, and give you an easy readout. And they were able to recover the genetic material, even though the virus was in those bodies, but the humans hadn't had the virus. So they got out the genetic material and they sequenced it. So they now had the genetic information. And then they went one step more. As we mentioned last week, you can take a sequence of a virus, the genetic information, and then you can turn it back into virus. Easily done. Many labs can do it. You don't need to be rocket scientists to do it. There are literally thousands of labs in the world across the globe that could do this. And then in 2005, they published a report in the very prestigious journal Science. We haven't talked about journals, but there are two journals, Nature and Science. These are the top journals. Everyone wants to get a paper published in Nature or Science. So this comes out in one of the two top journals in across all scientific fields. And it just so happens that just before, I think it was a few days before the publication date, people in the US started asking, well, what's this all about? Are you really sure? I think it was a gentleman in Congress, the US Congress, who asked, well, what's this all about? And so very quickly, to shut down criticism or discussion, the Science magazine, along with the NIH, they found a gentleman called Paul Sharp to write an editorial accompanying this, saying why it was so important to have this information. The reason given, cutting an editorial down to one line, basically this would help in the making of further flu vaccines. Okay, so this was not just nice to have information, this was essential information. So you can understand if that was the reason for this, then go. The answer is, I don't think anyone can point, and this was, remember this study was in 2005, we're now in 2025. I don't think anyone can point to any publication, any vaccine, any study post-2005 where it was clear that they used this information to improve what they were doing. I don't think you can find it. Therefore, this was nice to have from a virological side, but you've pulled a virus, you've resurrected a virus that was gone extinct, naturally gone extinct, replaced. So you've done it through this genetic means, the power of our technology, and you've resurrected it. This is in some ways just like making a novel human virus, like the studies of Dr. Kawaoka and Fouché, who made human viruses from bird flu. Basically, they've made a new virus. They've made the world a more dangerous place. They say there were benefits, but when you look at them, or even if you give them 20 years, let's be kind and say, we'll give you a look-back period of 20 years. They haven't done it. So therefore, nice to have, but basically made the world a more dangerous place. And therefore, I think that was a bad decision. And on top of that, rogue states, crazy people could decide to do this if they so wished. And so this is a problem. Now, this was talked up. This paper was talked up. It became a sort of like a poster girl research paper. And I've noticed frequently that when in the infectious disease space, to shut down discussion, people say, well, we need it because it's going to help us make diagnostics, vaccines, and drugs. This is the trinity. And I've seen it. It's been rolled out for COVID. It's been rolled out in the gain of function, and in the case of the vaccine for this 2005 Spanish flu paper. As though vaccines, you can't, it's sacred territory. Stop. And the fact is, scientists are always allowed to ask questions, should be allowed to ask questions. And if someone doesn't like your question, tough. If your question is good, and it may be unfortunate for the person who it's asked of, but if it's a good question, it's a good question. Scientists are there to answer questions. I mean, there are lots of silly questions which don't need to be discussed anymore. The Earth is not flat. Move that aside. Science has answered those questions. But if the question's good, well, then the question's good. If someone is uncomfortable with that, they should not be uncomfortable with a good question ever.
So that's one example of an informational hazard, and that's one that I think a lot of people have heard about, and especially in light of one of the key drivers of those resurrection experiments, Jeffrey Taubenberger being recently appointed to the head of the NIAID. There's been a lot of controversy about the fact that he was involved in that, and certainly I've been very publicly critical about appointing somebody with a role in that to such an important position. But for the purpose of this discussion, you wanted to talk about a second example of informational hazards that's not as well known. And that was something called a retrovirus. So do you want to talk about what a retrovirus is?
Yes. Our genome's actually full of fragments of retroviruses. It's estimated that maybe 6 to 8% of the human genome has got vestiges of past viruses that got into our germ cells, either the egg or the sperm, and got into the material, and then derivatives of those. It's been well studied. It's fascinating area from a virological point of view, but it's not that important to our listeners, except for the fact that we have these endogenous viruses. Now, what happens to them is that they get degraded and they get inactivated, because if you had a virus in your germinal cells that would basically and that could grow, this would be recognized by the immune system. It would be knocked out, and this would be end of the lineage. So they get inactivated, and those that are maintained have defects in them, so they can't make virus. They are remnants of active viruses. And there's a particular virus, so we'll call it HERV-K. It's just a name. And there are many copies in the human genome. So what they did is they put all these sequences together, and they made savvy reasoning, saying, well, that mutation there That was probably the one that killed that one. But in this one, it's right here, and this is the one. And they used their brains to sort of see which were the mutations that killed these retroviruses. And then they said, well, now I know it's that I'm guessing it's this. If I make that whole thing with these errors corrected, I should be able to resurrect that virus. And you can say, well, what a hazardous guessing game. Well, actually it worked, and it worked twice. Two groups did it. One group in France in 2006, and they called their virus Phoenix. So they're basically, the name tells you they resurrect this is from the dead. This is a virus that infected germinal cells. They give a lot of discussion in the paper, lots of hardcore virology, fascinating details, but they couldn't have known, they couldn't have predicted. They were playing with fire because they were making a new human virus, and they had no idea of the consequences. At least with flu, you can say, well, Spanish flu, we knew it's a nasty piece of work. It killed 50 million. That is on record. But this thing, would it be just a wimpy little thing, or would it be something as dangerous as HIV, which, remember, is a sexually transmitted disease, but HIV doesn't get into the germinal cells. And this is totally uncharted territory. They had no knowledge of it. In the French paper, the very end, they don't tell us under what lab conditions they made it, but they say this should be done under there's a biosecure safety level 3. But they talk about this, but they did it. We don't know. They talk about the precautionary principle, but nonetheless, they did it. The second group in New York City, they did it. There's no mention anywhere in the paper of the biosafety level at all. We don't know what level. It was presumably done at the biosafety level of BSL-2. I'm guessing, no idea. But there was no mention in that. And they made this savvy game of thinking of the immune activation mutations. They chemically synthesized the whole gene, and they got out a virus. I know these two men, the PIs, the bosses who did this. They're very good scientists, very good. Thierry Heidman in France and Paul Binash at the Aaron Diamond in New York. Excellent. You look at their track records; these are hardcore, like Professor Kawoka and Professor Fouché. You know, these are not crazies. These are really hardcore. But it seems to be that they weren't thinking about this, and if they were, they should have told us very clearly that this was being done in BSL-3 conditions. The work from the US was funded by the NIH. That by the French group was funded by the equivalent of the US National Science Foundation. This is the French equivalent of the NSF. And they did it, and they got this. And I think when you look at the literature, it's science moved on. They've forgotten this. It's been done, moved on. People want to do other things. Binash's group did the same thing for a sort of a vestige of a retrovirus in mice. But of course, we don't care too much about mice, so no one's paid much attention to that. But they made the world, these two men made the world a more dangerous place without any discussion whatsoever. Since I've been working on the gain of function thing, there's only one scientist who I've spoken to even knew about these papers. It never come up in conversation. Just one gentleman brought this up. He said, Simon, do you remember that paper of Thierry Heidman? And I said, actually, yes, I do. But as I told him, you're the only person who ever has mentioned this to me. And they were clearly not as dangerous, but they could not have known. They could not have known. And this was done essentially under the radar, or if that people were asleep, they weren't being controlled. Now, the Frenchman was saying, well, the precautionary principle, we should be careful. So he was aware of this, but there's too much silence on this. No discussion, no prior discussion, which I think is not right.
so in the context of this as an informational hazard, are you saying that just the publications that exist that are, as you're saying, are not that well known, represent informational hazards because people can use that information now to generate those viruses in the lab and try to use them in some sort of nefarious form? Is that
Yes, I mean, I think these retroviruses that were woken up from the dead. The the scientists kiss of the sleeping. Well, we can't call her sleeping beauty, but the sleeping death because viruses cause death. Yeah, kissing the sleeping death. Um, um, they didn't spark attention because they're not as well known, and Spanish flu, that's clearly a much greater thing, and those would have drawn far more attention. I was just bringing them up to show that um, attention gets focused by the most obvious things, and Spanish flu, resurrected, woken from the dead. It's being done elsewhere in areas where no one could have anticipated. It probably would go nowhere, but we didn't have the slightest idea. This is a virus that infected germinal cells. Germinal cells. There's nothing more important in sort of the chain of human development is the germinal cells. Descendant, your children, your grandchildren, and so on. This is gets into those cells.
Should we not even talk about this now if people are not aware of it? That's kind of what I'm getting at, is the fact that these papers were not well publicized a good thing? And so should I not even put this out? Is by talking about it, are we in fact amplifying it?
That's in the public. It's in the public domain. But I think the thing is, no one would be interested because we don't know if it's linked with disease. People who want to misuse science, they want a big bang. Spanish flu, unfortunately, is clearly a big bang. Clearly, a big bang.
But the idea of resurrecting retroviruses from taking human sequences, right? So sequences of humans that, and you can just look for these retroviruses and then do experiments to try to, like you say, raise them from the dead. Those sound like, I mean, as great, if not a greater threat than digging for viruses in the
I no, I
that way.
no, I don't want to go down this talk. I mean, in the infectious diseases, you sometimes see people trying to say, "Well, my microbe kills more people than your microbe, therefore my microbe
Oh,
is more important. Therefore,
I
you
see
should
what you
find my microbe and not yours." I don't well because that's the sort of conversation you would ensue. Um,
I get it. Okay.
Making these things which are the world was made a more dangerous place, and this occurred without discussion. In the 2005, there was no prior discussion of this. And making vaccines, as I said, okay, let's say that it would help. It didn't turn out, and it's not clear to me that would have worked. Just as it wasn't clear to me. Well, it was clear to me that those 2012 papers on bird flu, they could not help us in the making of drugs and vaccines and predicting the next pandemic. That was just rolled out to steamroller it through. It was done. Don't ask me why it was supported. I think, let's say that people didn't see clearly. Let's be kind and say they couldn't see clearly. But once it became public, we still had pushback. That is what you want. The mistake was made. Nobody's perfect. We know this. That wonderful film with Stan Lemon, absolutely gorgeous, Marilyn Monroe, and Tony Curtis. Anyway, fabulous film. But nobody's perfect. But once you've realized you've got a problem, handle it. And what we see is brushed under the carpet, brushed onto it, move along, and there's nothing to see here. That is the same sort of mistake as in COVID, when how to handle COVID pandemic, very, very difficult. A new virus, no one really knew what to do. And therefore you can say mistakes could be made in the phase. But as soon as you realize that some of what was being done wasn't right or wasn't effective, you can then change and make sure that you can get it and improve your efforts. So once if you see there's a mistake and you don't improve, you don't correct the errors, then that is the big issue.
Why wasn't there more attention to those papers that came out with this Phoenix virus?
People don't want to bite the bullet. They don't want to address the downside of science. Everyone wants to talk up the good side. So say you and I are an institute head of some research institute that receives money from an infectious disease, NIH, Gates Foundation, you name it, whatever, WHO, everything. And then we get some good news that we've got a guy who might found a vaccine to say something like West Nile virus, something like that. And so you talk it, the paper comes out, you give interviews, whatever. Everyone loves it, and people say, "Bravo, excellent." Now, someone sends in that same institute published a paper on a gain of function of a flu virus, and instead of making it infectious to the lung, some flu virus would give conjunctivitis, and this one gave very severe conjunctivitis. And some journalists got hold of this information. Well, as a institute boss, you want to sort of put it under wraps because you are a private organization. You survive through grant monies, and you don't want to show your donors and the people who give money to you that someone did a damn silly experiment. So the natural reflex is to push back, say, no, move along.
But this ties into the overall thing that we are trying to perhaps accomplish here, which is to overall change the culture of scientists and directed towards virology. Because, as you point out, there was no discussion about whether these experiments should be done before they were done. So you just get these, results that emerge from scientists that made a unilateral decision to place the world at risk without any input from any independent body to say, whoa, like you say, this shouldn't be done, or we should do this. We should think very carefully about whether there are any benefits or that could emerge from this to outweigh the potential existential risks.
Yeah.
So, and in both of the episodes of In Defense of Virology we've done, you've talked about something, I guess, a very simple way that perhaps the culture around, research could change, and that is if scientists would agree to a Hippocratic Oath. So do you want to talk a little bit more about that?
Yes, I think this leads to it. And as listeners know, the Hippocratic Oath, every medical student, when they take their thesis, I was there when my son graduated. He's now a cardiologist, but he got his medical degree. When he gives his defense of his thesis, after that he gets his hand up. He says, "Yes, I take the Hippocratic Oath," and I can't remember what the words was, but basically, first, do no harm. That is it. Do no harm. So that doesn't mean, when you think the bar is incredibly low. It doesn't say yes, I have to do everything to save my patient or to treat my patient or get this patient back home. The thing is, first, do no harm. A lower bar you can't imagine. That just be, don't be crazy. Don't kill your patient. You're not. No demand of results. The culture of of productivity. You say, don't do harm. And this goes back ages. My son and my wife are both medics. For them it's obvious. But for a PhD, me in virology, I have actually thought about, since gain of function, I have thought a lot about doing a black experiment, and I could do it. I thought about it. I know a number of things, and I wouldn't do it because it would make the world a more dangerous place. And that's what virologists and infectious disease people, from the time of Louis Pasteur onwards, they've been beating the hell out of microbes. And the last thing you want to do is suddenly start making new ones. We've got such a long list of things we want to do. The list of boxes unchecked, I mean, you've probably got 30, 40, 50 big boxes on big things which we still haven't done. And therefore, I just don't see this, and I don't think our job is to make the world a more dangerous place. Therefore, I would say, particularly for infectious disease, but maybe, let's say, all the life science students, when they graduate, they should take a Hippocratic Oath. Now, you can come in quickly and say, yes, Simon, but the gentleman who resurrected the Spanish flu, he was probably 40 years old when he did it. He's way past his thesis. And how can you can't ask him to take a new thesis and Hippocratic Oath. Right, this is a very simple solution. Let's start with the NIH. We'll move on to other people. Just the NIH. When the NIH finances a grant, it weighs up the quality of the research, and people say this is a really excellent idea. We're going to finance. Somewhere in the small weeds, the small wording, there's the paragraph says, every member of this proposed team promises to do no harm. We will not make the world a more dangerous place, if you wish. And if by inadvertence we suddenly find ourselves in an area where we suddenly realize that by totally unanticipated result, we've made a virus that is more dangerous, we stop unilaterally. We go to the NIH, we discuss, and we have to realize that because this is making a novel or a dangerous virus, that we just put an end to it. We put it under wraps and we just kill the project, and we kill anything we've made. This is part of not making the world a more dangerous place. As I say, the bar is incredibly low. Everyone should be able to step over this bar, and we haven't been able to do it. The NIH could just put that little paragraph, and you could discuss the wording. Some lawyer will have to get their or in, of course, because of whatever. But you got a little paragraph in there. Then you could do that, and then you could get control over people who are 20, 30, 40 years after their thesis. But you could get maybe the Gates Foundation, because it's a philanthropic organization, totally independent of the NIH, and other philanthropic foundations who've been financing biomedical research for years and years and years. I mean, did you realize that resurrecting Spanish flu or making novel viruses is in complete opposite of everything the Gates Foundation has been striving to do, and the MacArthur Foundation, and all these other foundations who have been using donors' funding? It's madness. Making novel things, it contradicts the whole ethos of these foundations. And if there was, as I say, we did in last week, if there was an upside, but a very strong downside, we could discuss. But as we saw for the bird flu, there wasn't even an upside. So I think this could be done. And the final part of it's actually three-pronged. There's the NIH, there's the philanthropic foundations, and then there are the universities, because there's a whole new generation of students coming through, and every year there'll be graduation ceremonies in summer, early summer. And at that point, you could, big universities, Stanford, Harvard, Princeton, Yale, but also those in Little Rock, Arkansas, and elsewhere, they could just say, look, morally, we think this is out. We're going to have a Hippocratic Oath for all our people, and I would say terrific. So you get the new generation coming through, and the funders get the generation who are post-thesis. It's not complicated. Who can be against making the world a more who can be in favor of making the world a more dangerous place? I don't get it.
We were talking before, this is an idea that's been around for a while, right?
Yes, if you look at the literature, people have been asking this, and it's not just life sciences, people thinking technology, engineering, and of course with artificial intelligence, you could say mathematics. because the genesis of algorithms, and looked at all this debate over the impact of Facebook on young teenage girls, and the algorithms keep making sure that you get fed the stuff that is very anxiogenic sometimes.
It shouldn't be that hard to do. It just seems like and some might argue, well, these are just going to be empty words. But to me, this is part of an overall, multi-pronged approach to changing attitudes and changing the culture of biomedical research, which is the goal of something like this.
Yes, and freestanding media, we have phenomenally hard tasks ahead of us. For example, treating cancers. There is no big one method that will probably hit 20% of cancers. No, it's all, well, this is a drug for ovarian cancer. This is one. You have the Keytruda from Merck, which is pretty good. But actually, when you look at the data, you realize that those on Keytruda, they often frequently relapse after 1 year of treatment. We don't have a magic bullet for cancer. And so we have a phenomenally hard problem up ahead of us, or we don't have a vaccine for HIV. We have big problems in infectious diseases. And there's this little thing saying, do no harm, an incredibly easy option, sign, done, tick. The bar is incredibly low, and we can't even pass that. That your listeners understand we haven't got cancer whipped, or we haven't got Alzheimer's whipped. They get that. They understand that. They know it's a damn hard problem. But Hippocratic do no harm. It's so low that that, and you can't even do that.
And it's arguably performative, right? So this is like an easy win for people in positions of power to implement. And critics could say, oh, this is performative. But the reality is, who cares? It's a nice, it's a gesture in and of itself that could have a positive impact. It comes with no cost. No cost.
And it shows the way. It shows that yes, we are concerned, and that science, and as everyone knows, science has downsides. I mean, if you can drop missiles on the enemy, well, the science behind that, nuclear bombs, the science behind that, and so on and so forth. We know all this, and this is easy. Why make new viruses? Why make the world a more dangerous place? It's a nasty place already. Look, there's the Ukraine war, but you're dragging out these. Why? And we can't even, the infectious disease people can't get through that, and the life science people can't do things. I can't speak about the science, the technology, engineering, and maths, because it's not my area. But certainly for life sciences, it's a no-brainer. We don't allow cloning of human beings. No. So do no harm. And it shows the way. It's a gesture. It's an easy gesture, so something can be done. And as we know, we've got to handle this dangerous gain of function. And it's not, despite the executive order, it's not over, as you know.
And the last thing I'll say on this is that, in fact, a year ago, both of us were on a panel at a conference organized by the current NIH director, and you spoke about this. And later on, I recorded a podcast with him. This is Jay Bhattacharya, as well as the current FDA commissioner, Marty Makary, who was also at that conference, in which they both seemed to be very enthusiastic about this idea.
I kind of feel like that's part of the training. You don't get ethics. You're not ever asked, what is the impact? Like, does this experiment have any impacts that might adversely affect others? There's this idea that the Bryce and I have been popping around with Simon Wain-Hobson about this Hippocratic Oath that scientists could take, right? Just as doctors take in principle should take an oath to say that they will first do no harm to their patients. Scientists should take an oath if they're moral, that to say that the scientific work that they will do, they will not do where they will that the work they do will not harm the people that they're studying or harm the public generally at large.
My wife is a pediatrician. My son is an interventional cardiologist. They've taken a Hippocratic Oath. they could do harm. But as a virologist who's thought about this, I could conjure up some really nasty stuff that you really wouldn't want to know about. And no Hippocratic Oath. So I think a Hippocratic Oath for life science PhDs, pretty easy, fairly easy to do. Stanford could start it tomorrow if they wanted, I'd imagine.
I mean, that kind of idea where there's this like ethical obligation to scientists to sort of think about the broader impact. I mean, often it's really hard to predict, right? You have some scientific advance, you don't know exactly how it's going to be used. That's understandable. But you're working on something where you stick something into a virus to make it more transmissible and deadly to humans. I mean, come on. What could possibly go wrong?
You did speak with Jay about this last year,
Yes.
correct?
Yes, but just, and this is where mea culpa, the Europeans haven't done it either. It's not an American effect. The Europeans, the Japanese, the South Africans, the South Americans, the Australians, no one's done it. It's not an American problem per se. We're talking about it because we were both at Stanford together, and this is something that concerns both of us. But my colleagues in Europe, I've mentioned the Hippocratic Oath to a few colleagues in France. Do you think they jumped on it? No. Do you think they jumped on it? No. No, we talk about the Scandinavians as a way forward and showing their civilized way. Did they have a Hippocratic Oath for life science students? No, they don't. So it's across the university science enterprise. It's not just an American thing. So if that reassures some of your listeners, I hope it is bigger than that. And I think the universities has been resistant because they don't like people sticking their oars in their areas. And it's this thing. People like to talk up the good news, and when it's bad news, they hunker down and they try and push it under the carpet because they think this will hurt them, and it's totally stupid. This, if people realize that the first university that takes the lead and has a Hippocratic Oath, people will be clapping, and there'll be editorials in the Washington Post and New York Times saying, "Bravo, you've got some." Well,
look, I'm gonna immediately after this. Well, not immediately after this. I'm gonna prepare an op-ed that will be, I'll try to pitch to a local newspaper to encourage Rutgers, the university that I'm at, to be the first to adopt a Hippocratic Oath, to take the lead. I do what I say, Simon. You know that. So I'm going to do this.
I do. Yes. And you are persuasive. And
well, let's not say that, but I will do it. But
you got me into writing these essays. You got me into doing these videos with you, right? As I said, I've said it before. It's your fault, and I'll back you up in your errors.
Well, I appreciate that. All right, so that's good. So look, we all have calls to action right now, or at least I do. I now gave myself a call to action: op-ed on Hippocratic Oath, directed to Rutgers.
If I may say, if listeners agree with us. Please think of writing to maybe a university in your city or your state. This can only help. Or if you want to write a letter to your local newspaper or whatever, yes, this will help. I think it will help.
Right. And it's easy to do, and it sort of seems obvious. Why would somebody not want to sign on to say, let's do no harm. Let's not make the world a worse place.
Yes.
I'm not sure.
And as we're going to see, we are listeners will hopefully learn that Bryce and I are very practical people. So in next week's video, we're going to come up with another simple request that should make the world a slightly more safer place. So you're forewarned.
Well, and with that, Simon, I certainly look forward to recording the next episode. But I'd like to thank you once again for joining me on Science from the Fringe's newest hit series, In Defense of Virology. My name is Bryce Nickels. It's a pleasure to be joined by Simon Wain-Hobson. Take care, everybody.